here in the middle, or in Chapter 8, you're going to learn about that genetic code that we just looked at is useful because it's going to come back and haunt you maybe here. But remember your 6th, 7th, 8th grade biology classes, you probably talked about this famous monk that's named after me that grew peas, and he noticed some of the peas were smooth and some were rough. Remember that guy's name? Yep, Gregor Mendel. And so he figured out that every time he took a pea that was smooth and crossed it with another smooth pea, he almost invariably got smooth peas out of it. But if he took 2 rough peas or wrinkled peas and he crossed those plants that arose from those wrinkled peas, he would almost always get wrinkled peas out of it. But if he took a wrinkled pea and a smooth pea and he crossed those 2, then he would get a predicted proportion of plants that produced wrinkled peas and of plants that produced smooth peas. And he basically described what is termed inheritable genetics, that genes are inheritable. If you're blonde-haired, blue-eyed, and you marry another guy that's Scandinavian, he's blonde-haired, blue-eyed, you're probably going to have blonde-haired, blue-eyed kids. And the same thing if you're dark pigmented and you marry somebody with dark pigment. In fact, if you marry somebody with light pigment, you're going to probably have kids with dark pigment if you are. And so he started describing what we know now to be inheritable genetic traits. And it's predictable. Mendel said it was predictable. Everybody else said it was predictable until the 1940s, 1950s. Barbara McClintock comes along and she said, wait a minute, it's not predictable. She was looking at, it's Thanksgiving time, so this is really appropriate. She was looking at that Indian corn we call maize that has all those different beautiful colors of kernels. Some are black, some are lighter blue, some are darker blue, some are yellow, some are kind of pink. And she was looking at those and trying to figure out, why are those kernels different colors on the same cob? She really wanted to understand what was going on with those kernels. And so she would take the dark kernels and grow up plants from those, and she would take the light kernels and grow up plants from those, and she would do a whole bunch of genetic crosses and cross plants that arose from dark kernels with other plants that arose from dark kernels. And she would do the same thing with plants that arose from the light kernels. She'd cross those with light. And she kept seeing patterns that weren't like Mendel said they should be some 100 and few years prior to that. She went to meetings, she talked about her results, and everybody said, this lady's crazy. It can't be this way. It doesn't happen this way. She must be making it up. What's wrong with this lady? And pretty soon, she just kept doing her results, but pretty soon people ridiculed her so much in the scientific community that she stopped attending conferences. She stopped presenting her papers. They wouldn't let her publish anymore because she was seeing something that had to be made up, something that couldn't be real. And so she kept working, and the problem was that everybody else in the scientific community believed that DNA was stable. But Barbara was seeing something that was very different from that. Finally, in the 70s, 2 guys saw something that looked very similar happening in bacteria. Okay, we progressed 20, 30 years, and now culture's different. Maybe women can be right sometimes. Sometimes you still got to wonder. Yeah, sometimes it's all the time, right? And so, but we got 2 guys, and they see something that's happening in bacteria, and they say, well, this is really weird. We grow these bacteria. They can't make this gene product. But if we grow them long enough, they can make the gene product. And if we isolate those cells that can make the gene product and grow them some more, pretty soon they can't make that protein anymore. What's going on with these guys? How do they do that? And they started dissecting what was going on, and they figured out that in that bacterium there are little bits of DNA that can jump from one place in the chromosome to other places. And when it jumped into the gene that they happened to be monitoring, it killed that gene, and it didn't work anymore. But those little bits of DNA could very precisely jump back out again and move someplace else in the chromosome. And when they did, they jumped out and they brought those splice junctions back together, and now that gene many times worked again. It made the protein that it originally coded for. And that's what Barbara was seeing. What she was really seeing, now we call these little bits of DNA, we call them transposons or transposable elements. Sometimes they're called jumping genes because they literally do jump around in the chromosome. My lab, when I was doing my PhD, we worked with a different organism, and there was something that was strange going on in that organism, and we figured out that one strain of that species that we were working with, actually, when you mated it, as soon as it got to have sex, the transposons got really excited, and they started jumping all over the chromosome and disrupting all kinds of genes. And we figured out, hey, that's pretty useful. We can knock out all kinds of genes just by letting this guy have sex. And so it's kind of like a fungal STD, because as soon as it has sex, a lot of them start dying, and then you can figure out why they're dying, because these genes start jumping throughout the chromosome. Not only can some of them jump from one place to the other, when they're in the process of jumping, some of them actually replicate. So now you've got 2 or 3 or 5 down in the cell instead of the original one that was jumping around. And so you can get more and more of them. This is really important, and we'll talk about why, but now you've got the idea that bits of pieces of DNA can move around. That's what Barbara was showing. That's what these studies are showing. And now you can put it together with the idea that we've got a problem in our society. That problem in our society is that we've got lots of organisms that are becoming resistant to antibiotics. And we just a few minutes ago talked about how in your gut you've got 1,000 different organisms that are living in your gut. Some of them are living, but some of them are also dying. What happens to their chromosome when they die? It gets shed out into the environment, right? And cells around it possibly are going to use that DNA for nutrients. Some of them have a way to pull that DNA into their cell and actually stabilize that DNA down inside of their cell. And now you've got thousands of different species worth of DNA getting dumped into an environment where there are lots of organisms that are sometimes being starved and are thinking, okay, I need a meal, there's some DNA, I'm going to take it, maybe I can use it for energy. And they pull anything that they can into their system, and sometimes that anything actually helps them a lot. That's exactly what happened with the E. coli 0157. Remember that guy? It's the pathogenic E. coli. I told you before, you've got E. coli living in your gut that doesn't do anything to make you sick. It just lives there. But this E. coli 0157, it's and we still have to deal with those all the time. In the U.S., vancomycin is one of the last resort drugs, and these Staph aureus are still resistant to vancomycin, or still sensitive to vancomycin. And so we can use vancomycin, but the only problem is there are lots of bugs that live in the gut that are also susceptible to vancomycin. As soon as we hit a person with vancomycin, we wipe out some of those organisms that are susceptible. And there's these little guys that live in your gut that are called vancomycin-resistant enterococci. They live in your gut, they're already resistant to vancomycin, and as soon as you wipe out their competitors, they grow up, and the outcome for the person who gets VRE is not very good. Many of them die. And so treating with vancomycin is not good because it can select for resistant staph that's resistant to vancomycin, but there's other microorganisms that are in the gut and in the body that can cause equal or worser disease. So when we think about organisms changing, becoming resistant to antibiotics or getting new characteristics, we have to think about how that happens. And it all goes back to what we learned about in the previous chapter, this natural selection process. You do it in your body every time you take an antibiotic. If you decide to drink cranberry juice because it's a beautiful drink and you love the flavor and you've heard it can make you lose weight, you're applying natural selection to the bugs that live in your gut and in other places in your body just by putting the chemicals that are in cranberry juice into your body. You're adjusting the bacterial populations, and those that are there try to adjust to those new circumstances. There's some diet, it's a fad diet, but it's called the Firehouse diet, and basically you drink Tabasco sauce for every one of your meals, and it does a good job of giving you diarrhea, and you mix it in with some other things, and so you flush out all the bacteria, and then you don't have the bugs to help you break down the stuff that you're eating, and so you don't absorb any of those nutrients. And go look at it, tell me if it works for you, because I thought about it, but I don't like Tabasco. So anyway, so I know there's a lot of ridiculous diets out there. I like this one. There's one out there. You can go read about it. If you deep breathe all day long, what's it do to your metabolism? Boost up your metabolism. And the reason your metabolism is not working as well as it should is because it doesn't have enough oxygen to run all those electron transport chains that want to run. And so if you deep breathe, you can actually burn more energy. And so try that one before we meet together and let me know how it works.